Hey y'all, happy Wednesday. It would have been tax day, but we're not having taxes right now. Tax deadline has been extended. It's pretty rainy here. It's made it nice to do work because I've been in and I don't want to go outside, but it makes it depressing because then I have to walk the dog in the rain. So there is 1 week left of classes after today, which is both amazing and depressing. So let's see. Today we're going to talk about a couple of other parts of molecular techniques. We are going to talk about bioinformatics for a little while. It will not be the same depth that we would have done in class or an in-person class, but we'll talk about what it is and some of the different tools. We'll also spend a few minutes talking about how to look at figures. So it's a little after 4:30, and I see there's a handful of you around. So if you have questions, pop in. But I thought we would first talk about our daily announcements. So today your homework 3 was due. It looks like I'll be able to grade those pretty easily through Canvas. So we will keep up to date and kind of keep in constant communication about that. The other thing is, if you need an extension, if something's gone wrong, send me an email. If you sent me an email right before this, I just haven't had a chance to respond to that. So hopefully we can all make sure our technical difficulties, there are none. So today there is another figure analysis opening. This one's a little different. For this one, you will need to read the paper and then answer the questions, unlike the other one where you just need to look at the picture. It's a really well-written article that I think is pretty interesting. So there's that. And then the other announcement, you guys are likely starting to get curious about your final and what we're going to do next week. So what I can tell you about the final is it will be run through Canvas. It is so the final exam is on the 29th. I think there's a typo on the typo's either on Canvas or on the syllabus, and I don't know which one, but it is on the 29th, the in-person one. Ooh, Christine, that's a good question. I want to say it's due Monday. Yeah, it'll be due Monday. So you'll have the weekend to finish that up. And then that will be the last assignment that you guys will have. So this is the last one. This last figure analysis. There will be no more homeworks, no more anything else. So you'll have that. And then the parts of the final. So it is, I have not made decisions about what we will do for our final. But what I can tell you is that it will either be, it doesn't really matter. Those decisions will at least be presentably made for the Friday YouTube live class today. We will have YouTube live class on Monday. Next Wednesday, to give you guys a little bit of an easier method to ask questions, although if you would prefer this format, so if you watch this, not live, like later, and you would prefer for the question and answers to be live, you can go ahead and do that. The downside is if we don't have any questions, then I'm not going to sit on YouTube Live and just talk to myself. I mean, I can, but that's not very fun for anyone. So I think the last class we will do either a partial review or something like that on the 22nd. So we have class today and on Monday, and then we will have a review either on Zoom or on YouTube Live, and then we will get the final after that. So that's kind of the schedule. We're getting to the end. It's happening. So before we start talking about today's material, does anyone else have any questions, thoughts? I don't want to say issues because it's hard for me to fix anything on YouTube Live, but thoughts, questions? Doesn't look like it. So today we're going to do two things. We're going to talk about parts of the paper, kind of a general structure of how to read them. And then we'll talk about what is bioinformatics, and I will provide you with the handouts that I would have provided you, but we're not going to actually use many of the programs just because it's really hard for us to try to do that at the same time. So when we think about literature publication, so many of you, if not all of you, have started to write lab reports. So lab reports come in a series of sections. And so we know that you have an abstract, you have an introduction, and the rest of it. And so how you write a lab report is different than how I, as I don't want to call it a grown-up scientist, but a scientist in the field, reads a paper. So when we think about a paper, it's important to think about what they are and where the information is. So every scientist has a different opinion on the order in which to read a paper. For me, I will, if I'm not sure if the paper is relevant, I will skim the abstract. So the abstract should function when you write it with a movie trailer-esque. So when you watch a movie trailer for a movie, it doesn't tell you the ending, right? Your abstract should have a one-liner in there that says like what the results are. So abstracts are a little bit less important in the digital age, but still super important, right? If you go to PubMed or you go to other websites, other repositories, the point of the abstract is what you see first. Okay, yeah, I want to keep reading this. So when I read a paper, I start with the abstract. Read that. Then I go into the introduction. So anytime in the introduction, when you come across words that you don't know, chemicals you don't know, proteins you don't know, I tend to read mine and either write my questions like, what protein is this? Or what does it do? Or I sit next to my computer and I look up those things. So even today, as a PhD scientist, I still use Google a lot when I read papers. Maybe I don't know what that reagent does. Maybe I don't know how it works. But the point of the introduction is to give you the outline of where the field is today. So in your lab reports, a lot of times you guys write what the technique is and what it is. In a literature paper, the introduction, the groundbreaking part of that is to tell you what the problem is, what we're trying to solve, and what do you do with that. So that's kind of what's in the introduction. Then comes, so the method sometimes comes after introduction or is at the very end. I will admit, I skip the introduction. No, I skip the methods 100% of the time. So methods, in my opinion, are really great if I want to recreate your experiment. But if I'm just reading to see what you did, I don't always need to read the fine details of how many microliters you added, what was your concentration, etc. So the method section has a point. But if you're trying to see what was the study, it does this section. I try to read that so in the results and discussion and conclusion sections, I try to write that. And so, or I read that, and then in the margins and underneath, I underline things that I think are important. I also will try to ask questions like, well, what is this? And then, so as you read that, when you get to the words figure 1, figure 2, figure 3, a lot of times we've unfortunately trained you guys, based on the way textbooks are written, to skip every picture, right? The pictures are not always as important as what the words say. In a paper, in a literature paper, it's almost the exact opposite. The figures themselves are the parts that are super important. So the figure allows you to go in there and say, where is their data? And so when we look at a figure, you should look at the caption, and then you should look and say like, okay, here's what we see here. You should be able to interpret that figure and hopefully get the same conclusions. So a figure, you know, I remember being in school and them saying a figure is worth 1,000 words. Not always true, but a well-developed figure gives you the idea that, okay, all of these things are happening. And so you should be able to read the data and the figure caption in order to come to a conclusion, okay? So you also want to make sure that what they've said about the figure is what you got out of the figure, right? So when you think about a biochemical assay, if there's a Western in the paper, you should be able to see the bands that they're talking about in the paper. You should be able to interpret that. So when I read a paper, write all over it. I write, here are the results that I see, here's what I know. If I go look something up, I will draw an arrow to it and tell me what the definition is. So as you read through the results discussion and all of those figures, you should be looking into all of those different sections. So the conclusion section is kind of like if you were really pressed for time, like you had to tell me what was in the paper in 10 minutes and you'd forgotten about it. In those 10 minutes, you should do the following. Read the abstract, look at all the figures, read the captions, read the conclusions. That gives you the overarching theme. Now, you are correct that if I gave you a 3-minute summary of any movie, you would have missed most of the details. But you would know the theme, right? So if the movie is, I don't know, A Day in the Life of Lily, and I said she had peanut butter on her breakfast snacks, and at dinner she ate 3 cookies and took naps all day. It's pretty accurate. That's what she did. For the record, the peanut butter is not because she's spoiled, which she is. It's how I get her to take her meds. But I gave you a theme. I didn't tell you that from 9 to 10:50, she took a nap in the living room, curled up in a Lily donut, and then we went for a walk, and then we did something else. I just gave you the highlights. So you'll miss some of the details, but the overarching theme is true. So there are other parts of this that I think are worth kind of looking into when we think about reading papers. So the other parts of reading papers that you want to do is you want to make sure a part of your job as the reader is to make sure that you agree with the conclusions that they made. It's possible that you don't interpret the data the same way. Maybe you get to the end and you have more questions. Not necessarily questions of how was this experiment conducted, but if it's about a protein, you might want to know, like, but what if you had done something else? It's okay for it not to be a complete story in the literature. Complete stories are often hard to get to. So part of your goal in reading of a paper is to determine what their conclusions are. Sometimes it's hard, sometimes it's easy, but you want to be able to see connections. And so I don't think I have any running around on my desk here. There's a bunch in my office that I could have shown you. My UNF office, not my house. I tend to write all over them. I underline things, I highlight things, I draw arrows when there are important facts. So for this figure analysis that was in homework 3, so in the figure analysis for homework 3, one of the things you guys, several of you stopped by my office hours to ask, you wanted to know about the C18 NTA. So C18 NTA is actually, like, if you don't know what it is, there is no shame and no harm in going to Google and typing that in. So C18 told you that it was an 18 carbon, and then it has an NTA molecule on the end. So it intercalates or inserts into the membrane. And so you could see that both in the figure and in the captions. And so one of the things that we really want you as scientists to develop, and this is why we're going to do this figure analysis, what if you don't understand something? There are a lot of times that I read papers where I'm not sure that I follow. And so there's nothing wrong with looking at the internet, looking at what paper to something else. So if there's a paper 1, you can go back and look at that. You can also do what I like to call a reference hunt, which can get a little bogged down, where you look and you say, okay, they said this. Well, where did that come from? It's in the introduction. It should have references. And so a lot of times if people say it and I'm like, well, that's interesting, I will go and I will read that other paper. Not in the middle of this paper, but I just developed this repository where I go and I read papers. So when we think about the primary literature, which are papers, the things that we want to be able to do is to analyze their data by looking at the figures, and we want to be able to read it and figure out all of the things. So the experimental context, the methods section, I have a friend, that is the first thing that they read. They open the paper, they read the methods. I don't. And the reason I don't is because I don't necessarily need to know all of those details if I'm not going to recreate the experiment. But sometimes when I read the results and discussion, there are things where I'm like, but how did they do that? So then I'll go back and look at that, whether the methods is the after introduction or at the end. And so different journals require different formats for how you make subsections or other things. But those are the overarching parts of like reading literature and kind of looking at those things. Does anyone have any questions about the role of different sections, how different sections work, other tips and tricks on how to read literature papers? Doesn't look like it. If you get stuck when you're reading a paper, feel free to reach out. Feel free to pop into my office hours. Feel free to look at other things. And I know that we're all afraid to use Google. And like, we really do tell you guys, like, never use Google. Don't. I use Google all the time. Like, what is this? How does that protein work? What does that protein do? It is just another repository of information. We do want to use our scientific skills to make sure that we are not using, I don't want to say unqualified sources, but we want to make sure that we're actually using a qualified source. So if you have other questions about that, let me know. But the next thing I think we'll talk about today is bioinformatics. So bioinformatics is a really broad term that basically says we want to look at the information within biological systems. So 100 years ago, when we were back in class, we talked about how you can talk about identity matrices, right? So we said you could create an alignment where two things align, and you can ask, are they exactly the same? And so an identity matrix is a really great way of asking, do you see the same residue in the same place? The thing with bioinformatics is we can start to look for both patterns So what I was saying before it buffered was if you have a positively charged residue, it doesn't really matter which one you have. Now, we as chemists know that there's an additional carbon in there, and that will change it from maybe a T-Rex arm to a longer arm. But the chemical interaction there is probably the same. So bioinformatics is going to give us the ability to look at whole regions and say, do we think this part of the protein and this part of this other protein could do the same thing? So in the way we are going to do that is you can make an alignment. So alignments we've talked about before, and we've done them by hand, where you basically say, okay, I'm looking for matches. But what about if instead of just looking for matches, you could use a computer algorithm, which is like super nice, right? You can use an algorithm and basically it will look and play with the alignment to see, does it align together or not? So we've talked about alignments for two species, where you take two things. And we did a couple of things where maybe you had 2 or 3 different sequences, but they were all small. Bioinformatics allows you to align proteins that are huge. And you can do the whole thing. And you can identify regions where like, okay, just these 90 amino acids are the same. And so the ability to do that kind of stuff is really important. So in my research group, we look for proteins that are similar. So proteins that are similar allow us to kind of ask questions like, hey, we think this protein looks like this other protein. Are they alike enough? And so there are a bunch of values. So I'm going to go get the whiteboard and draw some things up there for you. Whoa, sorry about the earthquake. Okay. So bioinformatics, sorry. Is the study of information of biological systems. So when we think about bioinformatics, one of the things that we often want to think about is what data is there? So we can talk about the genome sequence. We can talk about protein sequence. So within these systems, and they're all housed in a National Institute of Health database, and there are a few other databases, more and more today, they're all trying to be gathered under the same umbrella. And so the NIH is doing actually a really excellent job of reorganizing that system right now to make it more streamlined. So if 80 people have E. coli genome sequences, you can all put that data in there, which means if you go in to ask for something about E. coli, you're going to get 80 responses. That's a little overwhelming. I really just want to know, like, classically, does E. coli have this gene? And so we can start to ask designed questions. So we can ask things like, does this protein have a homolog? We're going to call this general. So you can do something called a blast search. And so blast searches is a specific database. And for the life of me, I cannot remember what that acronym stands for. But we can basically say, are there any similar? So you can ask, are there any similar proteins, general? Or you can ask a really specific question, which we'll call 2. Does this genome, you guys still see that? Yeah. Have this, yeah, you can see that, protein. And so the reason you would be interested in that, because I think that's a really common question. So why do you care? Does this protein exist? So if we think about any protein of interest, so we'll use lysozyme. So lysozyme has a job. It degrades certain sugars, peptides. Lysozyme degrades peptides. Yeah. So the question is, if we find it in hen eggs, do you also find it in eggs of other bird species? Do they do the same job? Do we only find it in the eggs? Well, do bacteria have a similar enzyme? So we can start to ask those questions. And so you can ask these in different ways. So bioinformatics is a series of ways that you can do this. Now, there are 2 or 3 different ways you can be a bioinformaticist. 1, you can design the tools to help people ask new questions. So you can analyze huge data sets. You can look for new information. So that's an option. 2, you could collect data. I tend to think about bioinformatics as a tool, a way for you to gain other information about systems that you can't look at with your eyes. So if I want to know how many of you want to eat chocolate chip cookies? Today, I would have to ask each and every one of you, do you like chocolate chip cookies? Do you like chocolate chip cookies, right? If I were to tweet, how many of you like chocolate chip cookies? I might get some responses. I might not get any responses. You never know. But when we think about this, bioinformatics allows us to pull everyone in the system or ask specific questions. So part of the tools we can do is we can run different types of alignments. So we can make alignments. And so alignments can have a variety of functions. One of their goals is to start to ask questions like, are these two things similar? So earlier this semester, we looked only at identity matrices. So an identity matrix, so we'll write this out, ID. So identity, if we have an alanine and an alanine, you get a check mark. If you have an isoleucine and a valine, you get an X because they are not identical. Now, based on what we know about amino acids, isoleucine and valine, they're both hydrophobic. They're roughly the same size. So maybe instead of a check mark, maybe we should get like, that approximation wiggle, like, they're kind of similar. So what about if we start to think about something that tells us more about this? So they're similar. So there's a way, there are a variety of different matrices where we can say, how similar is an isoleucine and a valine? But what about if we think about methionine? That's hydrophobic. It's not the same. And is alanine and isoleucine the same? Right? They're both hydrophobic. So you can start to get these scores. So when we talked about gap penalties, like eons ago when we talked about similarity, gap penalties become more of a point so we can have a similarity or homology scores. So they tell us about two proteins. And so these homology and similarity scores start to tell us things like, hey, well, lysozyme and other lysozyme are similar. But if we were to discover a new species where we have a sequenced genome, it might be advantageous for us to be able to look at what we can find in the genome and start to answer questions. So today, COVID-19 is why you're at home and I'm at home. So if you followed the news, which try to stay away from the news, but one of the things that has made this is there's a wealth of scientific discovery occurring right now. But one thing that is quite intriguing is how quickly they were able to discover the sequence. So early on in this pandemic, scientists were able to discover the sequence of this protein or of the genome. So we sequenced the viral genome. Now we've gone on to identify all the proteins that it encodes for. And so scientists are able to say, we've seen a protein that kind of looks like this somewhere else. So if it kind of looks like this and we think it might do this job, maybe we know what to do with it in this new environment. So if I gave you today an alpha helix, maybe it's a little bit different than anyone you've ever seen, but you would know it's a spiral protein. The residues are on the outside. Okay, that's great. If I gave you something totally different, it's a little bit more complicated. So things that bioinformatics are really good at are the ability for us to say what's happening in this region and what's not happening in this region. So there are matrices. So they're called homology matrices. So probably one of the more famous ones is called the Blosum Matrix. So Blosum matrices were developed many years ago, and so they come with different numbers at the end. So Blosum 62 is the matrix that I prefer to use. And like many of the things that I'll say, it's likely because someone I know was like, this is the way that we do this. So Blosum 62 gives you a score. And in what I will post on Canvas later today or tomorrow, soon, you'll be able to see that it tells you different things. So if you've never looked at a matrix like this, it has all of the amino acids. And so I'm going to draw a fake one. AW. And so you get identity scores. So A and A, we get a 1 here. That means they're the same. But if we have an A that switches to a W, so an alanine to a tryptophan, those are not the same at all. So in this case, we're going to take off 10 points. So when we think about mutations, this is often about evolution. So if an alanine evolves into a tryptophan, those two residues they may or may not do the same thing. Now, if it's in a hinge region, like some elbowy part of a protein, maybe it doesn't matter what's there. But if it was in the tight pocket of an active site, and you take an alanine and you replace it with tryptophan, it's feeling a little claustrophobic in the active site, right? So we know that's here, but an alanine to an isoleucine, I'm going to give you +4. Give you 5. So these are roughly the same. But an arginine to an alanine, I'm going to give you just a -2. Because it's not that bad. So from here we can see these are the same. And it goes in and it gives information. Now, you don't need to memorize this. I'm just giving examples. I have analyzed by hand using a matrix, but most of the time it is a drop-down menu on a blast search or something else. So this isn't something where people tend to do it by hand usually. Now, I say this to you, but I've never taught from a house before this semester. So maybe we're going to go back in time. I don't think so. But my guess is alignments are still going to be something that you can use online. So what you would do is you would create a score where you look at your two aligned proteins. So you have an aligned protein. Where you have 1 and 2, and you would look at every single residue and sum up. You'd sum up all the scores. Now, typically, on the internets, you get a homology percentage. What percent similar are they? So the percent identity and the percent similarity are similar numbers, right? So you get points for being the same in both matrices. But in a similarity or a homology matrix, it gives you a similarity score. So if we have regions that are similar, like the active sites are slightly changed, or we have long hydrophobic stretches, but we're using different orders of amino acids, that doesn't mean that they're going to do different things. So we can now start to say, okay, this region we think does this. This other region out here, we don't know what it does. It doesn't look like something we know. So the homology scores tend to have a slightly higher threshold. So when we talked about identity, I said that the protein needed to be somewhere between 25 to 30% identical, which feels really low. But the similarity score usually needs to be higher than 50%. But sometimes you can start to manipulate those numbers if you're looking at specific regions. The active site of the protein is here, and we see really high similarity, 80 and 90%. But the proteins are different sizes, so sometimes the similarity percentages are a little bit off. But the homology matrix gives you that ability. Now, in the algorithms, it gives you a percentage. When you do this by hand, it's really hard to determine the denominator, right? So a percentage is a top number, which is your score, divided by your possible score. So it is very difficult to determine that number by hand. So typically if I were to ask you to do that, you would then just report it as a score. So, but the homology matrix basically tells you what information it gets. So there are different matrices, and different matrices tell you different information. So there's the Blosum 62, the Blosum 50. I believe there's one in the 40s. Different scientists have developed them in different ways. And so that would be one aspect of bioinformatics. So where the matrices come from for the purposes of Biochem 2 at UNF, not all that important. But there are ways that you could say, like you could make an argument that an alanine to an arginine really should be a super high positive score. I think that going from a hydrophobic to a charged amino acid probably isn't that great. But it's not different. Like, it's hard to say. So when we think about these matrices, we can start to ask or answer questions about how similar these items are. And so you can do this. And so there are two different ways that we can look at this information. So you can create, so the types of alignments. So a pairwise alignment is somewhat like it sounds. We're going to align two sequences. We're going to look at two sequences to see if they are similar. Okay? So a pairwise alignment is just two. And so you can do something called a global alignment or a local. So a global alignment looks at all of both proteins. So a local alignment is somewhat different, right? So when we think about a global something versus a local, we know that a global is going to be a bigger picture and a local is very specific. So a local alignment looks for regions of high similarity. So if we were to ask two different questions, so a local similarity, sorry, I almost fell out of my chair. Happy Wednesday. So a local alignment looks for tiny regions within the protein, 10 to 15 amino acids, that are really high identity. But having 10 residues that are highly similar to something else in a 400 amino acid protein, that does not mean that they're the same. If you look at the global alignment, and there is a lot of identity, or a lot of high similarity regions, that tells you more about how those proteins may or may not function. So for example, how far down can you guys see? If we align two proteins, we are going to have the blue protein and the black protein. We're going to make this one a little fatter so you guys can see it. Yeah, good enough. So we have these two proteins. They are not yet aligned. So once we align them, if we complete a global alignment, let's just say they look like this. We have our black So we have these two proteins aligned in our global alignment. So when we did this, we can see that there's high scores, or we have lots of similarity here. Yeah, you can still see that. Regions of similarity, which is really great. But it doesn't actually tell us if we think these two proteins can do the same thing. So the problem is we have to know a little bit about the protein. Because if right here in the center, where my finger is the active site for the blue protein, they do not do the same thing. But if the active sites are if we have active sites here and here, so red square, active, then it's likely that these two proteins are just evolutionarily different. And so the question becomes, what does that tell us? A local alignment maybe would only give us information about one region. So I don't really know how to draw this, but the local alignment might only pull out this region. So oftentimes a local alignment doesn't give you as much information about how the two proteins are different or similar. It can give some. And so that's really good. So that is one type of an alignment, local or a pairwise alignment. Two proteins, we can align them and ask, what do they look like? So the other, depending on what you're trying to do, the part of alignments that can be better. Better is not a good word. Let's go with different. So a different type of alignment. So another type of alignment is something called a global alignment. Nope, let's call it, we already talked about globals. Let's call it a multi-alignment. Some people call these by the website name, so they'll call them a cluster alignment or a clustered alignment. So let's say you have 10 genes, 10 proteins that you think might be kind of similar. So you can look at pairwise alignments, but hypothetically, let's do a little back of the envelope, side of the envelope math. If I have 10 proteins and I want to know how similar they are, I can do 10 pairwise alignments. But that only tells me how similar protein 1 is to the other 9. Okay, so that's 9 alignments. But I want to know how similar number 1 is to 2 through 10. And I want to know 2 to everybody else. But I also want the ability to look at them overall. So a more global alignment of proteins. Say more than 2. More than 2 sequences. QQEN for regions of conserved homology. So one advantage to doing a clustral alignment is that I can now look at 15 sequences. I could align them all in the same way, and then you could start to look for regions that are the same in all of them. So maybe if we looked at 10 different sequences, and they look like this. So this is just 6, 1, 2, 3, 4, 5, 6, 7. And we need one more, like this. So I know that you're like, what is she trying to tell us here? These are our 10 sequences. So the thing is, it looks at all of them. Now, it's easier to look at in the program because it's not a series of lines. But what we can see is that a lot of our proteins, but not all, are lined up right here. This probably tells you that all of these proteins have something in common. So this would be a region of similarity. So, but we could also say that it doesn't look like very many proteins have this region. But we can also see that a pairwise alignment between some of these might not have given us that much information. Now, why is that? So when you have proteins that are of vastly different sizes, 100 amino acids versus 700 amino acids, even if you overlap perfectly, that's only a 1/7, which I think is roughly, I'm pretty sure on that actually, a roughly 15% identity or similarity. So it looks underweighted. So sometimes in these, it requires additional skills that you develop over time, the more interest you have in how this works. So these are 2 different types of alignments that you can do. And there are different, one of my friends always calls them the toolkit. Like, here are the different tools that you have in your toolbox when you want to look at different types of bioinformatical analysis. So these are alignments. So the other things that we can glean out of bioinformatical analysis is some other, what I would like to call, informational data points. But before we start talking about this, does anyone have any questions about alignments? So Ryan asks, what is the benefit of doing a local alignment? Personally, I've never used one. A local alignment can be nice if you think you have repetition. So if you think that your protein is concatenated, so a local alignment would be good if you have a protein. If this is protein 1 and protein 2 looks like this, but if we see a local alignment can be very beneficial if what you see in the difference between protein 1 and protein 2 is that protein 2 has 4 repeats of protein 1. So protein 2 contains 4 repeats of protein number 1. So that would be one advantage of a local alignment. Local alignments tend to be used if you are looking for specific identified regions because they tend to be quite small. But good question. Other questions? Excuse me. So the next thing we're going to talk about is what, I don't want to say like the social security information for a protein, but the information about a protein that is quite important. So if you go to the protein page, so in the NCBI database, which is where PubMed is housed, there are different websites for every protein. So if you go to a specific protein's website, so we're going to call it protein 5. On this website, you can determine the organism, where it came from, the number of amino acids, and it will give you a protein sequence. So a protein sequence is a series of alphabetical characters in a row. Lots of them. So knowing that your protein is 283 amino acids is great, but it doesn't actually tell you how big that protein is. What's its expected molecular weight? Count up how many alanines, and then add all those together. You could calculate the molecular weight of a protein using the same skills you would use in Gen Chem 1, right? How many hydrogens, how many carbons, how many nitrogens, how many oxygens. That would take a really long time. The other thing is we almost always think about proteins in terms of Daltons. Daltons is a unit that is not necessarily correlated. It's roughly, it is not the same as a gram per mole. Similar, but a little bit different. So one of the things is there are a number of websites where you can input in your sequence and get other information. So you could learn molecular weight. You could determine the isoelectric point. There are websites where you can predict post-translational modifications. You can predict the structure, or you can see if there are any structures. So there are a variety of websites and different databases where you can ask questions like, how much does this protein weigh? What's its isoelectric point? So isoelectric points allow you to do isoelectric focusing, where you can run a protein gel without SDS. So we talked about SDS page a while back, and I think someone asked, what happens if you don't use SDS? Will it run? Yes, but at some point it has to do with the native charges in the protein. So you could predict the structure, you could predict the isoelectric point, the molecular weight. There are ways if you think you have a membrane protein which we know that I care about a lot. But if you have a membrane protein, you can start to predict, does it have transmembrane domains? How many are there? So there are a number of different ways in all of these toolboxes for you to glean information about a series of sequences. Some of these proteins are predicted by modeling programs where we have a sequenced genome, and then they ask it, tell me about the proteins. So you can glean information about a protein that no one has ever expressed, purified, or looked at. So bioinformatics gives you the ability to really look into proteins in depth without having to subclone them, subclone, run Westerns, run RT-PCR. Now, there are other questions that you can't answer, and you will need to do that as well. But this is a nice way to learn about the protein really before you start doing things. So what other questions do you guys have about the information that I've shown you today? It doesn't look like anyone has any questions, so I'll hang out for just another second. But to be honest, today we just kind of want to talk about bioinformatics. On Monday, we will continue our discussions both of bioinformatics and other molecular techniques. You should expect for this to be somewhat heavily weighted onto the final since we've talked a lot about it. If you have any questions, you can pop them in the chat box. My office hours start at 5:45. I hope you guys are staying nice and dry today if you are around the Northeast Florida region or really wherever you are, because I think it's raining everywhere in Florida. But any other questions? Otherwise, I will see you guys on Monday. Stay tuned for more announcements, and I will post my previous year's bioinformatics handouts so that you guys can have them. Feel free to poke around on the websites and do other stuff. See you guys next week.